So, hello everybody. We have 6, 5, 4, 3, 2, 1, and let's start our course. We are extremely glad to have you here, and I hope that you solved all of these technical issues to have access to our course. And we are glad to start our day 0 or introductory session. Alexander, would you like
Yes,
to start?
that's truly amazing that we have such a global audience here today with us. And it's extremely great that we have more than 200 people just in a few days on the Slack, which is connected to the course. And thank you all. So today we will speak or show you the slides. And please feel free to ask us any questions you wish on the Slack or on the chat. And the third option on the YouTube, if you're watching us through the YouTube. We will try to moderate this all. And today with us, Marina, who is the initiator of the idea of such course, Dmitro, who will also help us with this course conduction. And my name is Alexander. I will also present myself a little bit later. So we have two big events for today. The first one is actually this introductory lecture where we speak about our course, where we will discuss our lectures, our programs, and more importantly, what you will get from the course. This will last for 1 hour approximately. And after this, we have prepared a networking session for all of you who wish to join. We have organized the breakout rooms where we can group up in up to 20 people, I think, maximum, or maybe 30, depending on how much of us will join. And there we will introduce ourselves, discuss our experience, discuss our practical issues or questions, and then we'll proceed to the course. So, Marina, microphone is all yours.
Yeah, so agenda for the first session today is to speak about the course, the main idea of the course, how it came to our mind, and also discuss the program that we will follow during next 1 week. Also, we will tell you about some issues related to practical part, because honestly speaking, we did not expect so many applications, and it was a pity for us to restrict the number of participants who will join the interactive practical part, but it was like necessary step. Another issue that we are going to discuss at networking is a networking opportunity, in particular today's networking session and also the networking session on the last day of the course. However, as you already experienced that we have this networking challenge in Slack, so feel free to write your bio or some announcement of the project you are working on. It's already a pleasure for us to read and know you better. Also,
so Marina, I have a question for you right now before we started. Imagine I'm the first year bachelor student and I have no idea what a single cell. Why should I listen to this course? Why single cell are important and why we are gathering here?
Yeah, actually, it's almost literally the thoughts that I have 2 years ago when I just started doing single cell, and I have no idea what is that, but I was extremely excited about the opportunities of this awesome method. So to be honest, we will discuss in details or like provide the answer on this question in details tomorrow. I will do this introduction to single cell, but in general, what you have to know is that single cell, in particular, single cell omics technologies is extremely hot topic of in scientific landscape, and almost everybody know and would like to work in single cell. It's my opinion just because I do work in single cell, but yeah, of course it's biased. So single cell in general is already 3 times method of the year from Nature IIIS journal publishing group. So it's really one of the hottest topic in current scientific landscape.
Yeah. So I couldn't add anything more. So I propose we proceed further and hopefully share some thoughts. Ask our participants to share their thoughts if they are also working with single cell. Because I totally agree, it's a hot topic, and I can barely imagine any biomedical field not benefiting from single cell approaches.
Yeah.
So
yeah.
what will we have on the course?
So just because we decided to share our expertise with you and in general make this information of single cell much more affordable, we decided to make this course, and our course that we're starting today is a 5 days course, which has 3 days of theory and 2 days of practice. Also, you will be provided with homework and a list of auxiliary resources that you might follow if you would like to succeed and continue practicing in single cell. To be honest, there are a lot of advanced materials for people who already have some background in bioinformatics or other transcriptomics related fields. So for them, this set of information is already prepared. And the idea of our course is to make information of single cell set much more affordable, in particular for people with a beginner level. So that is why, as you might know, after that, we are orienting on the people with almost basic background of molecular and cellular biology and not so advanced programming skills.
That's
So, yeah.
awesome. And also, you'll be receiving homework and additional resources from us. So after the workshops, we'll provide you more advanced questions or tasks for assessment. And if you wish to proceed, if you find this topic interesting, you can always share the results with us. We will evaluate them and give you some advice on either scripting, like we will make a code review, which we will do, and we'll discuss the results with you as well. So what is also important, that data which we use on this course is not our project secret data which we plan to publish. It's a publicly available data which we prepared for you specifically, and it's accessible to any person who would like to proceed with it. So it's not a secret data which we will not share with you. It's something shareable which you can continue work with. And with that, we'll proceed further.
Yeah, so because Alexander will be our tutor of practical part, he for sure knows better about needs and also requirements for for this interactive session. So thank you for this comment. Okay.
Yeah. So Marina, I think already has highlighted the background ratio for this course. And I very much like that we have very different or diverse audience. So for example, on the Slack, as Marina mentioned, we have the introductions channel. And on this channel, there is a lot of people who either never worked with R or CURAT or any tools for single cell RNA sequencing analysis, or already did something, but want just to see how other people work. And I think it's really, really astonishing and very exciting. So for me, it's also interesting, even though I will be mentoring some of you on the very basics, I also hope to learn something from you. And that's why every background matters here, and every background I hope will benefit from our course. We have already some questions on the chat. So the one question is, will the sessions be recorded and available? The answer is yes. During the course, all the recordings will be available on the playlist on YouTube. After the course is finished, we will hide this. So we will make some edits, we'll add some intros, we will just cut the non-necessary parts and re-upload them back. So in any case, every participant will be able to watch it in a few months or in 1 week after the course and still hopefully benefit from that. We will also discuss other questions as we go, because some of them are also already answered on the slides. But in the end, we will have a few moments to discuss the questions that we didn't discuss yet. So here we go to the analysis of our audience. So Marina, do you have any thoughts about the backgrounds we have today?
Yeah, like as people already introduced themselves in introductory channel, we have really very diverse community already. So in general, I just know that from our side, we will do our best to deliver the information and make it as much easier to understanding. So that's all I know. And I am pretty sure that even with a basic biology knowledge, you will be able to at least feel this way of doing a single cell analysis.
So there's also a question, what are prerequisites for this course? Do we need to have some background in the biotechnical field? So at least for the workshop, I will make very few slides with very basic explanation what we are doing and why we are doing that. So it will not be a problem even if you are from an IT direction, or if you studied something like computer science or mathematics, it still counts. And also, I think I've seen a few participants with non-related to biology backgrounds, for example, physics, And I still think it will be beneficial for them to understand the biological processes underlying these techniques. So this is something we also aim to discover. If you are a super experienced person in single cell sequencing, some of the parts may be boring for you, like to listen in 100th time what is a microfluidics or why we sequence cells in this way or not another. But I think for the majority of people, it will be something either to refresh or to get know better. And I specifically like that we have. I think it's less than 1%, but I'm not sure of people with advanced bioinformatical skills. That I'm really afraid because I cannot actually refer to myself as an advanced scientist in single cell RNA sequencing data analysis, because I'm working with data analysis of different biological data for 5 or 6 years already. But I started working with single cell RNA-seq only 1.5 years ago. So if you're people working with single cell RNA-seq from the very beginning, from the very first techniques, it would be very interesting for us also to hear about your experience. So here we'll proceed further, and we'll also read the questions. So what is also interesting, we were trying to understand what is the geography of our audience, like who you are guys, where you're from. And it's extremely amazing for us. Just take a look at the map. It's quite a lot of countries, I would say. And the majority of the participants were here we will provide this top 10 countries by the number of the participants. So even though they are personally from Ukraine, either originated or still working in Ukraine, we have collected the global audience, which is truly amazing. And well, that's all set here with this map. We're very thankful for joining us. And I think from this global network, we all can benefit, for example, by establishing new working contacts. With that, I also would like to thank our partner, the only and the single partner, the US-Ukrainian Foundation, and specifically to Christina Redko, who is the coordinator of the BioTech initiative. So thanks. We actually didn't expect so many participants because we have received more than 1,200 registrations, and our Zoom capacities were not quite ready for that. So thanks for the US-Ukrainian Foundation. We are prepared to host you all of you there, up to 1,000 people in the conference, and including the workshop, we'll have a lot of people and the networking sessions. So this is something we would like to mention and to thank our partners once again.
Just a small interruption, yes, because we have several questions in chat related to practical part in particular.
Of course.
So, people are asking if how can they access and how will they know that they are invited to practical part. So as you already have an instruction, you to join the interactive part, you have to fill the form, and then in next 2-3 days we will do the selection process, and till Thursday for sure you will receive the invitation and invited to separated Slack channel for the people who will join. And with all of us, we will make sure that you set it all of the needed software and environment to do this practical part with us.
So just to sum up, the registration form is still open up to 23:59 today, Central European Summer Time, which means that even after the networking session, you will still have a few hours to fill it up. It's really short. It's like up to 250 words, just so we're able to read this in some hours instead of some days or weeks, just to describe why they think it's cool for you or important for you and how your project can benefit from that. We aim to select approximately 30-ish participants who will then proceed to the interactive workshop session, meaning that they will join the Zoom session. They will receive immediate feedback if they have any questions. But for all the rest of people who will not receive the Zoom invitation, there will still be a YouTube link. So even if you are not inside the Zoom, even if we're not talking with you, like because it's physically impossible for us to host more than 30 people and still be able to answer your questions immediately, there will be YouTube stream and you can work in parallel with us. We will still answer your questions, but maybe slightly later, not immediately within the course. And of course, all the materials which will be used for the workshop on Saturday and later will be available for everybody. Means that you will receive the access to the scripts, you'll receive the access to the instructions, how to run the scripts, and still we will be happy to receive feedback from anybody. Like you have a question, write on Zoom, something doesn't work, still write on Zoom. on Slack, and we of course will be happy to help you. With that, I think it will be cool to understand who are the people behind the course, and I am extremely happy to introduce those who prepared this course. Marina, would you like to start first?
Yeah, so I would like to make a step back and discuss the idea of this course, how it appeared, because we will we will highlight it as well when we will talk about our organization in general. But in brief, the course appeared just after some long conversation between me and Vladislav Kalaka, who is absent today, but like he is one of the main organizer of this course as well, and your tutor of the second practical part. So this course started because the demand of single cell materials and educational courses was high, like from our perspective. And as I was in the genomics UA already, I asked to make some kind of comprehensive collaboration between us and build such a lovely team. So in brief, we are all from different parts of the world right now, but originally all of us are from Ukraine. Yeah, so as you can see our affiliations, most of us are PhD students, or Yulia already have a PhD status. So yeah.
So just to sum up from left to right, one of the lecturers and one of the people who are involved in the workshop will be Vladislav. You will see him in the further sessions. He is an MD PhD candidate, meaning that he is treating patients, and in between he proceeds with his PhD, and actually his project involves quite a lot of omics. So even for me, it will be very interesting to get know better what he does. Marina, who is the initiator of the whole project, of the whole course, she is a PhD candidate in the Netherlands, and also she will be one of our lecturers, and hopefully will also join the workshop sessions to correct us if we are making something not as she does usually, and of course it will be also interesting. The third person on the list is Dmitro, who is also here right now with us today. If he likes, I would invite him to briefly describe himself. But to sum up, he's a data analyst in the National Health Service of Ukraine. He is not independent. He is not directly involved in the lectures because of his work with, as you know, more emergent topics like pandemics. But still, we're very happy to have him in our team. So, Dmitro, the microphone is all yours. If you're here, feel free.
Thank you, Alexander. Nice to meet you all today here. So actually, Alexander described my works, and I just help to handle chats and other organizing stuff. So actually, that's all about me.
That's very humble, but we're really thankful to Dmitro, because actually a big part of this job and of gathering us all together here is his direct responsibility and success. So thank you, Dmitro, and we will see you on the following lectures as well. The fourth person on the list is Yulia, and I'm personally very glad to see her here, because she was working as a postdoc in Harvard Medical School for quite a while, and recently she has transferred to the biotech, to the industry. For some time, she was my mentor in the scientific meeting, so she helped me to proceed with my PhD. She was advising me on some questions, and I'm extremely happy that she will be one of the lecturers on the course. She has quite a lot of experience with omics, with single cell, and particularly with ATAC-seq, one of the hot topics as well on the field today. And finally, myself, my name is Alexander. I'm also from Ukraine, currently working on my PhD project in Vienna, Austria, in the Institute, Ludwig Boltzmann Institute for Rare and Undiagnosed Liver Diseases. My project is related to liver diseases, and here I also use omics, either public data analysis or preparing our own data within our projects to review and elucidate the specifics of liver microenvironment in different liver diseases. Partly our workshop will be based on this question, and we'll be investigating the destiny of immune cells which are involved in the inflammation and liver disease. So stay tuned, and with this we will proceed further to our program. So here I will ask Marina to comment on this program and to highlight us what will actually happen with.
Yeah. So as you can see, today we already started our first session, and the networking part is not highlighted on the slide, but anyway, you're like, we invite you to a networking session today. In general, the course will have 2.5, I would like to say, day of theory and 2.5 day of practice, because first day on Monday, tomorrow, we will make the introduction to single cell technology in general, and also talk about wet labs processes, which you have to make in order to receive your data to analyze. On the second day of the course on Wednesday, we will touch the topics of epigenetics and also immune cells landscape, some particular protocols related to single cell analysis. And on these 2 lectures, you will have the basic theoretical background on what's going on in these particular aspects. On Friday, it's my day. I highly recommend you to join these sessions. I will talk about tools which you can use to do single cell analysis without programming. So in general, these set of technologies and online software which you can use for free to analyze your data is already available. So that is why I will highlight many of them, also to make a stress on the basic instruments which you could use, or also databases, some website that you can use as an extra to make the most advanced single cell analysis. Another part will be on single cell multiomics, and it will be mostly theoretical. In our course, we will not do single cell multiomics practice, but yeah, as we will evaluate this demand from your side, maybe we will do this on our next session, if we will, I mean, if we will make the course in some time again. So next on the weekend, we will have the practical part, and it will start in the evening and approximately take like as every session from 3 hours. And we will on the first day and Saturday, together with Alexander, we will talk about and you will practice the basic single cell RNA sequencing pipeline. And on Sunday with Vladislav, we will do analysis of cell surface proteins. So actually that's all. And I just would like to recap that everybody will receive the instructions on how to set up your environment and also get prepared for this practice. But unfortunately, we are unable to fit to Zoom this interactive session, all of you. However, all of the notebooks and also all of this data will be available, and we will do our best to answer your questions and solve your issues if you will have them to help you get started working with single cell. So actually, even if you will not be selected to this course, you can do everything the same. So, and we will do our best to supply you with answers.
What is more important, we really will focus on selecting the equal representation, meaning that if you haven't ever started R or haven't ever started any script, it's totally fine. We'll try to combine the people who never worked with any bioinformatic related tasks with people who worked with something, maybe even with single cell data analysis, so it will be more or less balanced. So please don't be afraid of submitting the form if you are not an experienced person in single cell data analysis, but still would like to learn something. So already we have a question. Is the time on the schedule is Ukrainian time? So it is a CEST, which is Central European Summer Time, or Vienna time, Berlin time, Paris time, Rome time. It's all the same. Additionally, on Slack we have sent the link to the website where you can check actually what is the time, the course time right now. So it's the link is to the same time zone as the course. So we're very happy with that. And now, Marina, you will have a tricky question from me. Imagine you would be the first year bachelor student without any knowledge. What would be the most exciting course or lecture for you to participate?
Well, I strongly recommend to start with the basics. I mean, from tomorrow and follow the steps, because like the ideas and as how we designed that course, that by studying with us steadily, you will absorb all of this knowledge step by step. So actually, to start with, of course, all of these topics are extremely attractive, but to start with single cell, please make sure that you have a basics of understanding what is single cell technologies, what is the difference between single cell and other, we call them already bulk sequencing technologies. So yeah, if I were you, I would follow all of the lectures and then just trying to do my best to figure out all of the unknown terms, or also some concepts that I did not clearly understand. As a student, please feel free to join all of our session or watch the recordings if you are unable to join us in the real time.
Yeah, I think it's perfectly fine. And personally, I am interested the most in the lecture regarding the DNB cell receptor sequencing, just because I know it's also a hot topic, but actually never worked with that. So I am excited to learn about this a little bit as well. And I know that Vladislav actually works with quite a lot with this data and with such approaches. So I think we all benefit from the course. Thanks to Marina for creating this schedule and this opportunity for all of us. So here, I think Marina, you have put on the form to show how does it look for the practical part of the workshop. So it's super simple, just your email, so they can contact you further, your full name, and then just maximum 250 words. But really, if you would just put, I don't know, 50 words, it's totally fine, doesn't matter. Please describe your project which you are working on, or if you are not working on any single cell related project, describe the hypothetical project you would love to work on. With this, we will just be able to screen the candidates, again, considering the skill level they have already, and then to select the equal representation of people on the course immediately in the Zoom. And all of those who are willing to participate as well, but unfortunately didn't join the Zoom session for that, are more than welcome to participate by the YouTube stream and work in the parallel. And we still, of course, will accept all your questions, all your results, and discuss it with you in the personal manner or via the Slack channel as you wish.
Yeah, or in general, redirect you to already solved issue that have actually the same content. So we also will try to be as fast as we could, but in general, we all of us are full-time employed. That is why it could be problematic. So please take some time as well to wait to the answers.
Yeah. And also we have the question, if, for example, if some participants will be unable to attend the live session, how will their questions be answered? So for that, I would ask you, if you'll see us via the YouTube record, for example, or just skip some lecture, just write a question on the Slack channel. For example, today is day 0, as we defined that. So if you have any questions related to that, feel free to write it and we will answer even after the session or 1 week after, doesn't matter, whenever you see or read this information. So we will go next, and actually this is the presentation of our Slack channel. If you are not there yet, we have the I think I have the laser pointer. Yes, exactly. We have the day 0, 1, 2, 3, 4, 5, etc., which means that today we're day 0, the next day will be day 1. If you have the question regarding the next day's lectures, just post the question right there and we'll be able to answer. If you don't know where to post the question, just go to general or random and ask there, and we'll still find your question. Very important for both the networking and for the communication during the course. If you would like so, if you feel about that, please write about yourself a few words in the introductions on the introductions channel. That's important for us, so we can then understand what is the background or bioinformatical level of the participants, first of all, and second, we'll be able to communicate further. I have already seen that a lot of persons are looking for collaborations, and it's super exciting. So if you have listened to the course and you have participated in the practical sessions and you would love to start with single cell, just go to the introduction and see what people do. There is a great chance that we can actually communicate with somebody and ask to join to the project or ask to collaborate at some point, and it will be beneficial for your career. So we have
also,
already a lot of comments regarding the emails. So according to our email service distributor, we see that approximately half of the emails weren't opened, and we still are not sure what exactly happened with those. So either they just dropped to the spam folders or haven't been delivered at all. So for that, we're sorry, of course. And if you know any colleagues who have not received the email with the instructions, please write to us, connect us via the email, and we'll provide all the necessary links and the access. So for more than 1,000 of participants registered for the course, it seems that some of the emails weren't delivered. So we will try to fix that if you will provide us with the contacts. So we will go further unless there is something to add. I think no.
yeah, I think
Yeah.
no.
And also important part, we have the repository, the GitHub repository attached to our course. It is also published on the Slack, so you will find the link there. A few days before the workshop session, we will add all the necessary code and the instructions to the GitHub repository, so you can download it prior to the session and to evaluate it. How does it work? So we will separate our instructions to two parts. One of those will be for experienced users. Here we will just provide you with the data sets and the links, sorry, not the links, but the names of the libraries for R which you should install. The second option would be for not so experienced users yet. We will just provide you the image of Docker and also explain how does it work. So all you need is just to download one file, install one program, and run the file. After that, you will receive the access to the environment with all the necessary libraries or packages already installed. And all you need just to follow us during the workshop. So you will decide yourself which track is best for you. And if something doesn't work, we're of course always here. And yeah, that's no problem. And we have a very interesting question. So what some person is experienced with R, but doubt about ScumPy. So I personally named my session analysis with SUROUT, which is an R package, just because I use it most of the time. So if you're okay with Python, but not okay with R, you of course are you're totally allowed to use Python for the workshop. And even more, it's super interesting for us if you share the results and we compare them after. But my personal workshop will be dedicated to analysis with R. And I think for most of the users, it will be easy and beneficial to follow it because I think it's quite simple and straightforward. But if somebody is fluent with Python and would like to work with Python, of course, it's more than welcome. It will be provided into options, as I told, and the beginners options will be just a container containing all the environment you need. So all you will do is just to run two files, and in the result, you will open such a interface in your browser. It will be written in the instructions, and all the necessary code will be already there. So we will just go section by section with explanation, why do we run this code, how does it work, how does it look like usually, and in the end, we will try to interpret the results which we receive. This is just an example from one of the drafts we had and internally tested just yesterday. So a few days before the workshop, you will receive all the materials via the GitHub. And here Marina will briefly comment on the changes to the initial program.
Yeah, because as I said at the beginning of our session, that we were extremely excited and even surprised of the number of participants who decided to join our course. And that is why we decided to spread our program and include this extra day of theory and extra day of practical part. So actually we, as you have seen in the initial course description, we announced just 4 days, but then we decided that it's an awesome opportunity to share more knowledge and also make our networking session much more effective. So the first changes, the first set of changes is related to the schedule and the program. Another issue is that we already discussed that because of this amount of participants, we have to restrict the users for interactive sessions. So yeah, actually, I think we already highlighted all of these issues before. However, one of these forms that we kindly ask you to complete is related to evaluation of the lecture that you attended. So for example, starting from tomorrow, you will receive after each of this lecture and each of the session evaluation of where you will highlight your experience and describe how useful and relevant was it for you. However, our main motivation, and we are very open to receive the feedback on how could we improve, or maybe how to we focus on the other topics more to make our course much more useful and comprehensive in the future. So actually, this is a form that you literally have could contribute to the community and also make the course better for the next sessions, because if we will succeed a lot, we will continue for sure.
Yeah, exactly. So for us, it will be very valuable because it's definitely not the last workshop we prepare for you, and definitely we will prepare more, maybe on different topics in the future, and we'll of course be happy to consider you again and to evaluate. Maybe everything we will tell you will be super simple, and you will tell, no, actually know about this, and skip to the more interesting parts, or maybe vice versa, it will be super overcomplicated, which we actually try to avoid. So your feedback is more than helpful and welcome.
Yeah, and as I said, it will be after each practical day from tomorrow. You will receive the link to the chat related to that day, and also in general chat. When you can follow the link and complete this form. It's very short, and you will do it like less than in 5 minutes, like as I tried. So, yeah. And also, sorry, just a very repeating question about the recordings. All of the sessions will be recorded, including practical. So, yeah, you will not miss this information and tutorial, just in case that a selected set of people will be able to make it in real time to ask the question if they will have so. But all of these materials will be open access, available all the time.
Yep. So there's also a question where participants can get us in case of questions or the further courses. Yes, so the idea would be LinkedIn, but also if you stay on the Slack after the school, after the course, I will of course send off our announcements there. So the idea of the Slack is not to make it just for one school, but to make a collaborative space. I will tell a little bit about the organization and what we used, which approaches have we used in the past, and I will show you why it works in the best way like that. So again, the question regarding the environment, it will be uploaded to the GitHub repository a few days prior to the workshops with all the instructions you need. And we'll announce about that on the Slack. Also, people ask about the certificate of attendance. Unfortunately, we cannot provide that because we are not affiliated formally. I mean, our program is not affiliated nor accredited by any university. However, if any participant can benefit from the confirmation that he or she participated in this course, we're happy to contact the program of your choice and to write that you actually participated in that. And since we don't prepare any evaluations or homework for people not participating in the workshops, we again cannot score you or give you any marks, which means that the only thing you will actually receive from the participating in the course is one, the skills which you learn, the information about the approaches, the workshop skills, etc. The second one is you will be able to increase your working network while participating in our networking sessions. So that would be our answer. And here I will just briefly describe you the approaches or the history of our organization, how did we work before. So first of all, don't be scared. This picture was made actually before COVID. That's why the people are so close and without masks. So we have started in 2018 as a very small community for bioinformatics, for people who like to learn bioinformatics. And here you can see Yulia, who is one of the lecturers of our course. She actually, back then, I think it was December 2018, she was showing us how to make the differential gene expression analysis and how to plot all this. And it was very funny because we actually just gathered in a small library with laptops, and we have solved all the possible problems with installation of RStudio and R. And finally, it was a very nice experience. And here's our very small team after the first workshop. And it's incredible that actually today we have more than a few hundreds participants. And of course, it's very much, we very much enjoy this fact. So back then, we also started to make events in other cities, not only in Odessa, Ukraine, where we originated with a few participants, but also it's a picture from our lecture in the Kyiv Polytechnical University, which we made, I think, 1 year ago. Sorry, one year after the first event, and we saw actually that there's a lot of people, not necessarily from the biological background, who want to learn something in bioinformatics. This could be microbiologists or chemists, or, for example, physicists or computer scientists who actually would like to work on something biology related. And this is one of the ratios for us to host this course as well. Yes, and this is just another lecture which I personally enjoy very much up to this point. This is a picture from Marina's first try of the school, and could you please describe a little bit the background of this photo?
Yeah, I was reflecting on this photo today for long, because I remember clearly how I came back from my first traineeship in single-cell biology, which I did in Max Delbrück Zentrum in Berlin, and I was so excited and did not want to wait to share my knowledge and some information about this awesome technology. So we organized a 2 days course, like 2 days lectures about the basics and diversity of single cell methods, and also talk about application of single cell analysis in biomedical science. So yeah.
I mean, this slide looks very cool. I'm not sure I exactly understand what is it, but I like layers, I like different cells, and I like the plots. So I think it looks impressive.
Technology to know. Everybody should know the immersive fish, when you can visualize every RNA molecule during using this high resolution microscopy. So it's like
Perfect.
one of the, it's even subcellular resolution of single cells. So it's like.
So I expect there will be a lot of questions after this session or during your lecture about this technology, so be prepared.
Okay.
Additionally, what I would also like to highlight that we are not a local organization, because we actually have a lot of friends, partners, and collaborators from all over abroad, you can imagine. So I think it was in 2019 when we established our connections with Romanian Society of Bioinformatics. And it was something very new for us, because for us, it was the first let's say, formal, non-formal course we received as a team. We actually went to this workshop of Romanian Society of Bioinformatics, and we received this very structured knowledge about different things like alignment of the genome, like counting reads, annotations, etc. And I think partly, at least for me, it was something that inspired our today's session. And more recent meeting with global partners we had is a meeting with Next Step Science, which is a Slovak organization to spread biotech, bioinformatics in Slovak Republic. So that's also a presentation that we have a lot of collaborators from abroad, and we're very happy to include you to this network as well. And generally, I tried to prepare this map. So the people who are either providing lectures in this course or will participate in this course as invited, I don't know, people to provide workshops or whatever. So from our organization are now based in different countries. So originally we all worked in Ukraine, but now we're present in different countries. For example, Julia is working now in the US. Marina is working now in the Netherlands. I'm happy to provide you with a workshop from Austria, etc. And this light blue countries, highlighted countries, are the countries where we had invited lecturers in the past for our organizations, either in the workshop format or as lecturers. And we're incredibly happy about that. So if you represent any organization, or if you have a bioinformatics club, or if you have any colleagues in mind who could provide us with any experience-based lecture or show something from their own research, it would be incredible, and we would very much like to get to know you better in the networking session. So please do join and share your experience and the background. So I think it's very obvious why do we do networking, but still it's something we should speak once more. Marina, it's your turn, your slide.
Yeah. So I remember clearly when I was just graduating from my bachelor's, I was very skeptical on networking at all. So I was like, okay, I do participate in conference just to present my research. I even do not need any feedback. So I was like this toxic kind of person. But in general, now I realize that sometimes network is even more important than theoretical background that you can operate, because a network is a very powerful tool to spread your expertise or knowledge and also find new collaborators to build these new links and also highlight some uncovered topics of biomedical science or in general any field that you are working in. So that is why we highly recommend you to join as much network sessions, of course, like if you have a time for that, to interact and yeah, and improve your soft skill as well. So yeah, that's in
Perfect.
brief.
And it's very exciting that we actually have participants from Australia, and they write that there is 2 a.m. now. So I'm super sorry that unfortunately for you it's not the prime time, but still we're super happy to have you here today with us. And here we have prepared a small checklist for you. So for your convenience, what it's the idea to make to fully participate in the course. First of all, to join the course Slack. I think all the participants of this Zoom session have already joined it. However, if not, please let us know. Write us either on Zoom or YouTube. We will send the link at once. So if it's relevant for you, if you want to participate on the hands-on, please register to the workshop up to 23:59 CEST today. So just to sum up, you have still approximately 5 hours starting from this point until the deadline. And the form is super short, as we showed to you. So please feel free to write. Even if you have no prior experience, if you have no prior background for single cell, it's still very good that you would apply for that, and we would invite you to join it if it will be possible. So join the Slack channels related to the meetings. I think now it works automatically, meaning that you join the Slack workspace and you immediately connected to this day 0, day 1, etc. But still make sure that you can join this and you see the pinned messages, because these messages contain the link to the program and to the Zoom meetings, for example, this one. So people here, right, I don't know anything about the Slack. So basically the workspace where there is a chat and different channels for the workshop, for the school. Please refer to the link above, which one of the colleagues have shared with you. It will propose you to register and to download the app, or for example, to open it via browser, and then you can join the workspace and see all the links. So prepare to participate in the networking, which will actually start in 14 minutes, the first one. And also the second networking session will be on the last day. And I think it's super interesting to understand what people have learned, and or either whether the expectations have been pleased, so to speak. And of course, please do ask questions on Slack, do ask questions on the chat whenever you have anything you don't understand. It's technically impossible to consider all the backgrounds of you guys, but it's our main purpose and goal to make it available, accessible for you, even if you are not a biologist with specific focus on single cells. So please do ask questions, do meet new colleagues, especially in the networking. Do not hesitate to contact, to communicate with them. And take as much as possible from the course content, because it's something we would like to share with you. It's something we would like to learn from you as well. And I think it will be of our common benefit that we will communicate with each other during the course. Marina, do you have anything to add?
Yes, I have. I think I have 3 issues. So actually 2 questions in our chat looks interesting, because we highlighted and answered on them from different perspectives. So I just would like to recap all of the materials, including Docker container for installation, all of the instructions, how to set up environment. After the lectures slide that we used for our presentation, and the recordings of the video will be available. So basically all of this information will be distributed among GitHub, YouTube, and Slack, or in general, mostly like GitHub and YouTube, as everything related to theory and practice, and other is video supplying for that. And also, so we are not going to delete it, so you will have this access for for a long time. Another issue is also related to setting up the environment, in particular the RStudio and other packages. So everything that we will ask you to do, download or whatever, is open source and for free. So you definitely do not pay for nothing in this course. Our course is free to attend, free to run with us, to like run the pipelines and tutorials, and yeah, and we will spend our time to answer your questions as well, to support you for free. So yeah, and the third, yeah, I said three. Yeah, so the third issue is related to networking. As you can see, there are three of us from Genomics UA, and but we assume that we will split you in 5 or some other amount of breakout rooms. So there is a small chance that you will start without us. So please feel free just to start chatting or expressing your bio experience and yeah, introducing yourself each to one to other. So yeah.
So how it will look like right now? Right now we will have a small break after we will finish talking for approximately 7-ish minutes, and we'll start sharp at 19 or 7 p.m. test. So you will join the second Zoom link, which is connected to the Day Zero channel, which is already there from the very beginning. You will see the breakout rooms. We will distribute ourselves between 3 breakout Zooms, so please feel free to join any of them you would like to. In the beginning, we will ask you to introduce yourself, like in a few words, up to 1 minute, so everybody will understand who they're talking with, which backgrounds are present, etc. And then we'll just discuss different issues or questions regarding the single cell topic in general, about bioinformatics, about something very specific, and whatever you would like to. And even if you join the channel when one of us is not present, please just start introducing yourself and just feel free to speak whatever you wish to about. And of course, they're happy to receive your feedback.
Yeah, just try to keep it polite and in like a disciplined way, because if everybody will start speaking, we might not hear everybody of you. So yeah, just let's make it.
Yes. So the rule number 1: respect each other, and do not allow any hate speech or whatever, because we're all the colleagues here, even if not all working with single cells. Still, I think it's very professional and friendly community that we created today. So thank you all, and please refer to the Day Zero channel for the second link for the networking session.
Yeah, and if you have some questions, yeah, please leave them on the Slack on Day Zero or general, or you can write us directly, but it will be better to post it on the channel, because then we could reply, and then everybody will see the answer as well. So yeah.
See you in 8 minutes. Bye-bye.
Yeah, bye.